Okay, great. So I've been attending these meetings for a few years, so it's a particular pleasure now to be here as one of the speakers, talk about some work we've been doing in the intervening time. So this is about docking atomic models into cryo-EM reconstructions, and I'll turn on my pointer here. Yeah. Okay, so just to clarify what I mean by docking, you probably all know, but I'll just make sure. It's basically figuring out how an atomic model can be rotated and translated to place it into the right place in the map that you've reconstructed. So there's different things, different problems within this. One is the search problem. How do you explore all of the orientations and the translations to place the molecule in the map? Should you do it by a brute force 6D search, which is what a number of the cryo-EM docking programs do? Or can you separate the rotation and translation search, which is the approach that we've gone for, as I'll explain. And then there's the scoring problem. So once you've rotated and translated the molecule, how do you decide whether it's in the right place and score the right place from the wrong place? Very typical thing is to use map correlations with either a normal map or some kind of modified map. In crystallography, we've been working with likelihood for a number of years, and we wanted to explore that approach. So we use likelihood as a target. And then there's really the decision problem. If you get an answer, how do you decide that that's actually the right answer? And that's an important thing. How confident can you be in the solution? Okay, and then we also want to decide what is our target? What kind of problems do we want to be able to solve? So here's an example, beta-galactosidase map at 2.2 angstrom resolution. Now, we do want to be able to dock things into this easily and quickly, but if you can just stick it into Michelangelo and get an atomic model, the docking is kind of not as important. Okay, this is something that's a little bit more of a challenge. You're not going to stick a 6.9 angstrom map into Michelangelo and get a structure out of it. But if you know the structures of the components, and what I've chosen here is just to dock 2 copies of a small domain that's only part of the structure. That's harder, that's more useful. It actually turns out to be reasonably straightforward, which is nice. The problems that are most interesting so far and hardest are things like this. This is an E. coli complex 1 structure. So Janet Vonk was talking about other complex ones. This is the E. coli version. This is a particular map that we've been looking at where the resolution varies from 3.8 angstroms in the best part to something like 9 to 11 angstroms in the part I'm showing you here. And even with the model in the right place, you can hardly tell it's right. So the question is, can we determine from the fit of the model to the data that this is the right place for it to be, and can we find it? Okay, so we came into this from our background in crystallography, where we use likelihood as a target for doing a number of things. And the problem that is similar to docking is the molecular replacement problem in crystallography. And the reason we expect it to be useful as a way of thinking about it is that the diffraction data are in Fourier space, like the cryo-EM data, if you're trying to think about likelihood in cryo-EM. The big difference, of course, is that in crystallography, there are no phases, so that changes the nature of how you do things considerably, but the underlying features are similar. So in molecular replacement, we don't have phases, so we determine the phases by taking an atomic model and rotating it and translating it to put it in the right place. And then you can calculate phases and calculate a map. And this now, especially in the days of AlphaFold, is the way that the vast majority of protein crystal structures are solved these days. And we've put this into a program called Phaser, which does likelihood-based molecular replacement. And so we were interested in seeing what lessons that we learned in developing Phaser that could be applied to cryo-EM docking. Well, the first one is that actually likelihood is a really good choice. It has some really big advantages. Now, likelihood is a probabilistic measure. We're saying, what's the probability that I would measure this data set given my model? If your model is good, the probability will be high. And because it's probabilistic, it accounts for all of the sources of errors if you do it properly. So there's the errors in your data and errors in your model. It gives you an absolute score. The likelihood score is an absolute number. And that means that if you have alternative hypotheses, different models are fitting into the same data, the one that gives you the higher likelihood score is a better model. And even better than that, the absolute score, the number means something. If the number is high enough, you can be pretty confident that you're correct. So this solves this confidence problem. Another very useful thing is that before you actually do the calculation, you can calculate what likelihood score would I get if I put the model in the right place, given the quality of the model and the data. And that allows you to optimize the choice of strategy, and that's an important part of the docking procedure we've developed. Now, one thing that's different is that errors in crystallographic data are pretty straightforward. They're just random errors in intensity measurements. Errors in cryo-EM data are a bit more complicated and harder to figure out. So that's been most of the work involved in this. Okay, to assess the error in cryo-EM, you need to compare, or one way to think about it is to compare the signal power with the noise power. And the signal power is reduced if the sample can't be reproduced from sample to sample. So if you've got different conformations from one molecule to another in your particle images, and so on, you will reduce the signal power. The signal power will vary with position in real space because, maybe the bulk of the protein is all in one conformation, but one domain wobbles around, and then the signal power in that part of the map will be lower. If something's wobbling back and forth, the direction that it wobbles in, the Fourier terms in that direction are systematically lowered so that you get less signal power in different directions in Fourier space. So it varies with Fourier space in both resolution and direction. And then, of course, there's all sorts of effects like radiation damage and uncorrected particle motion. Noise power can be thought of more in terms of how frequently do you observe each Fourier term. And so not all Fourier terms are equal once you've got a whole data set. You look at one single particle image and you get a slice through the Fourier transform of the particle that's perpendicular to the orientation that you're looking at. Now, if you could sample the orientations uniformly, then you would get uniform errors in all of your Fourier terms, but because you have orientation preferences, you get massive differences in the redundancy. And what you need, so each particle image is an incredibly noisy source of data, and you need many observations in order to reduce the noise by averaging. And if your redundancy varies a lot, then the relative noise varies tremendously as well. So you end up with a distribution that's going to be very complicated, and it's certainly not isotropic. It's not just a function of resolution. So the way that we have settled on is to look in Fourier space we compare half maps, look at the two half maps as the source of information, and look through Fourier space in local regions and make local averages of the signal terms and the noise terms and calibrate the model that way. So the noise power comes from the differences between the half map Fourier terms. And these actually should be relatively constant over the whole map. And they should be reasonably reliable because there are differences between the half maps and their Fourier terms everywhere in Fourier space. The signal power then comes from the correlations between the half map terms. And those can vary tremendously over the map. So in different regions, if there's a lot of motion in a particular region of the map, the signal power will be lower. And also, the correlations, the relative error in a correlation is very high when the correlation is low. So there will be places, as you get to high resolution, where you really can't estimate the signal power very well. And I realized later, doing more of the reading I should have been doing, that this is something that Shor's dealt with in Rely On, that you have to have some kind of a model of how much signal you expect so that you can sort of do a Bayesian correction for the signal power that I was expecting to get at these regions where you've got low signal. So that's what we're doing in the signal power. This is, so getting back to the early slide where I showed you the three different docking scenarios, this is the third one, this E. coli complex 1. And in this case, there were Serious orientation preferences. So here these red spikes represent the preferred orientations. We're seeing those orientations quite a bit, which means the Fourier terms perpendicular to that are well represented. The ones parallel to that are poorly represented. This structure, as I mentioned, has a wide variation in resolution from about 3.8 to something like 9 to 11 angstroms. So if we look at this part of the map here, which is the part of the map that I showed you in that earlier slide, this corresponds to one of the chains, chain L of this protein. If we do this analysis for that part of the map and look at Fourier space in the same orientation as the real space, you can see the signal power is sort of anisotropic, as you might expect. The noise power is much higher in the direction parallel to your preferred orientations because those correspond to Fourier terms you're not seeing a lot. So it all makes sense, really. Okay, so we can put those together. Once we've got the signal power and the noise power, we have an estimate for each individual Fourier term. And we can then weight the reflections using something that we call diobs, which is basically a reflection or a Fourier term by Fourier term equivalent of the FSC ref, so the FSC to the true map. So it's computed with a formula that if you've looked at formulas for the FSC and how it arises from signal and noise, it should be familiar to you. And this number varies from 0 to 1, depending on the signal to noise. So if the noise, the error term, sigma E, is small compared to the signal, then the diobs will be 1. If it's large compared to the signal, very large, it'll be close to 0. So there's one program that we've developed called Prepare Map for Docking, boring name, but anyways, it computes the Fourier terms from the half map and these diobs weights. And this can actually, in principle, be used for any likelihood-based calculation with those data. It's published in this month's issue of Structural Biology, otherwise known as ACTCRIS D. Okay, so that is calibrating the error model. Then if we want to figure out what is the likelihood function for judging how well the model fits to that map, then we need to know also something about the quality of the model. And we can do that very roughly, and actually in these days of AlphaFold models, the errors will be pretty small. We can say if our model accounts for a certain fraction of the total content of that map, and it has a certain RMSD error, we can predict the quality of that model and combine that with the errors that would come from that with the errors in the map to get the terms that we need to calculate a likelihood function. And I'm not giving you the equations because if you're interested, they're in the papers, and if you're not interested, you'll just be bored. So, so then we've got the likelihood function. And then if we want to dock a model into a cryoEM map, there's this question of the search strategy. And we take the approach that's taken in molecular replacement usually, which is you break it into 2 3D searches. So you do a rotation search, you find the orientations that are more plausible, that agree with the data, and then a selected set of those are used for translation searches to judge the fit to the map. Now the rotation search, when you don't know the position of the molecule, you don't know the phases, so it just uses the amplitudes. That means it's equivalent to the rotation function we use in crystallography, and we could just reuse the code that we had for that. The translation search is, it turns out this likelihood target for the translation search can be evaluated exactly with a single FFT for all translations, because it's basically mathematically a correlation function. Now, the next bit I want to tell you about is how we can adapt the strategy to the particular problem. And we do that. So there's some strategic choices that we have to make. And that depends on the quality of the data and the model. And we do this through calculating something called the expected log likelihood gain. So I need a little digression to explain what that means. So first, what's the log likelihood gain? So step back, the likelihood itself is the probability of the data given the model, as I mentioned. And you can think of that as the product of probabilities for all of the individual measurements. Now that's, it turns out it's more convenient to work with the logarithm of the likelihood, and that turns the product into a sum. So it's a sum of logs of probabilities for independent measurements. Now you might be thinking the measurements aren't independent in cryo-EM because you've stuck a small molecule in a big box. And so you have an oversampling of the Fourier transform. But you can correct for that oversampling really quite simply. This is a suggestion from Van Hale and Schatz in a preprint from a couple of years ago. You just multiply by the ratio of the minimal box that's required to contain your reconstruction and the total box size of your reconstruction. And that corrects for the oversampling. Okay, then the third component is the gain, the log likelihood gain, and that is a measure of how much more do we know from the model we're using compared to a completely uninformative model. So if you just knew that you had atoms in there, but not where they were, a model with infinite errors, that's an uninformative model, but that gives you a likelihood score. You subtract it from what you get for your real model, and that gives you a difference. And that difference means something. 0 means you have a completely uninformative model. Something positive means it's better than random. And it turns out a score of 60 or more in a search usually means you're correct. So this allows us to decide whether we've found a solution. The reason this works is that the log likelihood gain is connected to the idea of information content. So it's telling you how much information in the data have you explained by the model. Okay, so that's the log likelihood gain that we determine in order to determine the expected log likelihood gain. In other words, the prediction of what we would get with a certain search strategy. Actually turns out to be relatively simple formulas. And we can do that knowing the quality of the map and the quality that we expect from the model. For the rotation search, that's the same as what we worked out for crystallography because it doesn't use the phases. And so we've got the code for that. We also have figured out the translation ELLG, but actually we don't need to know that because if you can find the orientation, the likelihood score you'll get from the translation search is much, much larger. And if you can find the orientation, it's easy to find the position of the molecule. Rotation is the hard step. Okay, so if the problem is going to be very difficult, as I'll get to in a second, you can actually make it easier if you knew where your molecule was. So let's say you're looking for one subunit in a big complex. If you knew where the subunit was and you just cut out that bit of density, you would have all of the signal that you're looking for and much less of the noise. So if you cut out a smaller volume, then you actually increase the log likelihood gain and the expected log likelihood gain, inversely proportional to the box volume. And here we're actually indirectly using the phase information because cutting out these little boxes, little subvolumes requires having phase information, which you have in cryo-EM. Right, so in our program EM placement that does the docking, there's a strategy that takes account of all of this. So first, it evaluates the signal and noise in the entire reconstruction. And then it says, if we searched over the entire reconstruction, would we expect the rotation search to succeed in giving us the right orientations? And if that's true, you can just run the rotation search and then do a translation search with the best peaks from the rotation search. If you don't think so, then it looks at how small of a subvolume could we cut that map into little subvolumes, and would that smallest possible subvolume be small enough that the likelihood score would be bigger, and it would be big enough that you'd get a success. Most of the time, then the answer is yes, divides the map into subvolumes and carries on doing a separate search with each subvolume. And if no, then there's a final fallback, which is to do a brute force 6-dimensional rotation and translation search, which hardly ever gets invoked. If potential solutions are found, then in order to put them all on a common footing, it does what we call focus docking, where you just cut out the sphere that's big enough to contain your placed model and calculate the likelihood score with a rigid body refinement. Okay, so there were 2 papers that were back-to-back in this month's edition of Structural Biology. And there's the second paper describes the implementation of this tool and some test cases that vary quite a bit in the resolution that we're looking at and the completeness of the model. I'm just going to focus on this one test case here, the E. coli complex 1. So I mentioned this subunit L out here. That actually turns out to be really hard to find. It's related to subunits M and N. These three subunits are all about 25 to 27% identical. But as you move along in this membrane domain, the resolution, local resolution gets better. So if we search for chain M, doing the search over the whole reconstruction, then it figures out what is the score that it will get in the log likelihood in the rotation function. It figures out 7.1 is a number where it says that's too small, we should cut out subvolumes, and the calculation says 4 subvolumes should be used. It searches over those, finds 2 solutions. They both refine to log likelihood gain values over 60. One of them is higher and has a higher map correlation. And it turns out the one with a higher score puts it in the right place. The one with a lower score puts it in the wrong place, but this is it's not random. It's a related chain. And if you search for chain N model, then you get a much higher score in this region, and you know which one is right for sure. This all happened in about 10 minutes, which isn't bad. If you search for chain L, it doesn't work because the map is so bad here that when it compares it to what it can do by putting it in the density for M and N, those are much better. Okay, so if we dock chain M, you can see the model in the original map. It also produces something that we call a focus map, which in this case looks a little bit better because it's taken account of the local signal and noise. You can see that it's not random, and you can kind of believe this is right. Okay, what about chain L? So we have another procedure called in place local, which takes advantage of the fact that if you can find exactly the right volume to look in, the search will be even more sensitive and it'll be much faster. And there's various ways of running this, but the easiest is to use a new plugin into Chimera X, which I'm going to show you in a demonstration in a second. This isn't in the current release of anything. So in order to run this, you need to download the 1.6 release candidate of Chimera X. You need to install a Phoenix release candidate because that's not quite there. In a few weeks, the official releases will probably work. And you need to install a plugin for Chimera X. If you want to do this, Dorothy Liebschner has put up some YouTube tutorials on the Phoenix channel of YouTube. You just look at the Phoenix Chimera X playlist. Okay, so let's look at this demo. Right, so this is Chimera X, and the scenario here is, let's imagine I've placed everything except for chain L. And I think this has got to be where my chain L goes. Right, so can rotate it around and see what we've got here. So the first step is just to center approximately on where you think you want to look for your molecule. There's my search model down in the corner there that'll be placed. And you choose under Tools, Phoenix, Local EM Fitting. This works with half maps. So, well, you choose the model and then you choose the half maps that you want to use for the docking. You can optionally say what the resolution should be, or else it'll guess. And then click OK. And it pops up an orange sphere that shows you how big the sphere is that it's going to be searching in. And that's a marker, so you can choose the move the marker around so that it's covering the right region that you want to search in. And once you're happy with that, you press Start Search. And then in this case, it takes 30 or 40 seconds to run. So what it's doing is first, it's cutting out the density in that region for the 2 half maps, comparing them, working out the signal strength and the noise strength to calculate the likelihood target, doing the rotation search, and then for the plausible orientations, doing a translation search. And then for anything that comes up as a peak, it does the local docking refinement. And finally, we'll move the model into the place that it's found, along with a map that you can look at for the likelihood weighted map. Okay, it should be done any second now. Yes, so here we are. There's our model, it's moved it there. And as we saw before in the earlier slide, you can hardly tell that it's right when you look at the density. But if you bring in the model that was deposited, and of course, as Janet showed us earlier, there are higher resolution structures of this, we know this is the right place for this subunit, and it's found it. Now, I forgot to mention the LLG score that's disappeared off the top here was 61, which is just barely over our 60, but it was the only high peak in there. The local map correlation is 0.3. So it's finding something that you can't see by eye because there are features that don't show up in connected density. Okay, so this is all available if you get nightly builds of software, but there's a lot of things to do still. So one really obvious thing is we haven't got a way of doing a rigid body refinement if you've already placed something, especially if there are multiple domains and you'd just like to adjust the multiple domains. That'll be easy. Have to do it yet. We're ignoring symmetry so far, so we can account explicitly for point group or helical symmetry in the search, which should improve things. And right now, it only does one component at a time, and it doesn't pay any attention to what you've already placed, which is something we can improve based on what we've done in molecular replacement. So you want to account for what's already been explained in the map and avoid clashes. And something I've talked to Stars about a little bit is getting away from the idea of deducing everything from the half maps and maybe seeing if there's some way of deducing more about the accuracy of the Fourier terms from the raw data. Software is all available. The prepare map for docking is in the CCP-EM library, which is very liberal license. Phasor TNG is one component you need to use. That's in Phoenix CCP4, will be in CCP-EM. Same for EM placement, and in place local is currently just available in Phoenix and Chimerix, but it should be in CCP-EM at some point too. And I just have my acknowledgements here. So Claudia Mian did a lot of the initial preparation of the EM placement script. Early built Phasor TNG that underlies things. Tristan had a lot of useful opinions on how to do this and how to do it in Chimerix. Tom Terwilliger told me how to do things in CCTBX, which if you've ever tried to use CCTBX, you need somebody's help. And Dorothy and Billy were involved in helping the Chimerix people down here to integrate the tool with the plugin. And Tom's been putting it into CCP-EM. Kathy Lawson helped with some of the searching of the EMDB. That's it. Thanks.